Hey everyone, welcome back to the Food Tech. If you're passionate about microbiology or just curious about how we see the unseen world, this video is for you. Today, we'll explore different types of microscopes and how they help scientists uncover the secrets of tiny organisms. So, let's get started. If you're looking for more in-depth notes on this topic, check out the link in the description below for detailed resources available on my PayHip page. Don't miss out on this extra content to enhance your learning. Before we dive into the different types, let's first understand what a microscope is. A microscope is an optical instrument used to magnify tiny objects that are otherwise invisible to the naked eye. It helps scientists, researchers, and medical professionals study microorganisms, cells, and materials at a microscopic level. There are various types of microscopes, each designed for specific purposes. Now, let's explore them one by one. Types of microscopes. First, we have the bright field microscope, the most common and widely used microscope in labs. Principle: uses visible light to illuminate the specimen. The light passes through the specimen, and contrast is achieved due to the absorption of light by the specimen. Best for stained specimens like bacteria, fungi, and tissues. Limitations: poor contrast for unstained or living cells. Next up, the dark field microscope. Unlike the bright field, this uses a special condenser that blocks direct light and allows only scattered light to reach the lens. This makes the specimen appear bright against a dark background, ideal for observing live, unstained microorganisms like spirochetes. Principle: uses a special condenser that blocks direct light and allows only scattered light to enter the objective lens. This makes the specimen appear bright against a dark background. Best for observing live, unstained microorganisms such as spirochetes. Example, Treponema pallidum. Limitations: low resolution and difficulty in identifying internal structures. Ever wondered how scientists observe living cells without staining? The phase contrast microscope is the answer. It enhances the contrast of transparent specimens by converting differences in light refraction into variations in brightness. This makes it great for viewing live cells, their internal structures, and motility. Principle: converts differences in refractive index of cell components into variations in brightness and contrast, allowing visualization of transparent specimens. Best for observing live, unstained cells, including motility and internal structures like organelles. Limitations: not suitable for thick specimens, as overlapping structures can cause artifacts. Now, let's turn up the glow. The fluorescence microscope uses ultraviolet light to excite fluorophores, making the sample glow in vibrant colors. This technique is widely used in medical diagnostics, such as detecting tuberculosis bacteria using aramine-rhodamine stain. Principle: uses ultraviolet light to excite fluorescent dyes, or naturally fluorescent molecules, which then emit visible light. Best for detecting specific proteins, pathogens, or cell structures using fluorochrome staining. Example, Mycobacterium. Tuberculosis detection by aramine-rhodamine stain. Limitations: requires specialized dyes and fluorochromes, and UV light can cause photobleaching. Finally, the powerhouse of microscopy, the electron microscope. Instead of light, it uses a beam of electrons to achieve ultra-high resolution. There are two types. Transmission electron microscope provides detailed internal structures by transmitting electrons through ultra-thin sections of the specimen. Scanning electron microscope provides 3D surface images by scanning the specimen with electrons. Principle: uses a beam of electrons instead of light, providing much higher magnification and resolution. Best for observing viruses, cell ultrastructure, and nanomaterials. Limitations: expensive, requires extensive sample preparation, and only non-living specimens can be observed. So there you have it. From bright field to electron microscopy, each type has its unique role in exploring the microscopic world. Which one fascinated you the most? Let me know in the comments. Don't forget to like, share, and subscribe for more science content. See you in the next video.